So generally speaking, most metabolic flux is controlled by a very few number of regulatory enzymes, and these will generally be the focus of the course. The ideal enzyme for regulatory intervention is one whose quantity or catalytic efficiency dictates the reaction it catalyzes and is slow relative to the other reactions in the pathway. So our cellular flux for biochemistry is really summarized here, where we have nutrients coming in, which are consistent with small molecules such as glucose, and these are used to synthesize larger molecules, or the smaller molecules are oxidized into even smaller molecules to generate ATP, which can be used as an energy carrier to synthesize larger molecules. Anything that's left over from this circle of metabolism is then produced as a waste. So to regulate the flux between nutrients, metabolism to small molecules, oxidation to smaller molecules to generate ATP, or synthesis of small molecules to generate larger molecules, can be determined by decreasing the quantity of the enzymes responsible for rate-limiting reactions, such as through degradation, and these will immediately reduce metabolic flux through the entire pathway. Conversely, an increase in either the quantity or the activity of the enzyme can force the reactions in a different way. So if we look at this general reaction scheme, you can see that the products of individual reactions could potentially feedback and influence the rate of downstream reactions. This is commonly referred to as feedback inhibition, and this could be both competitive, non-competitive, or allosteric. In this example here, you can see that the product B could inhibit the production of product C, and likewise, C could reduce the production of product D. This, of course, is a schematic illustration, and we'll look at very specific examples throughout the course. One specific example of feedback inhibition is illustrated here. When we look next at glycolysis, we'll see that the initial step in that pathway is the phosphorylation of glucose to glucose 6-phosphate. This reaction is catalyzed by the enzyme hexokinase or glucokinase, depending on what tissue you're in. The product of this reaction, glucokinase, can feed back and inhibit the activity of hexokinase. This reduces the enzyme's ability to phosphorylate glucose to glucose 6-phosphate. This is a classic example of feedback inhibition. In addition to feedback inhibition, enzymatic reactions can be regulated by the amount of enzyme present. And this is a regulation between the enzyme being degraded into amino acids versus the enzyme being produced or synthesized from amino acids. And this is a constant balance such that we can regulate product formation simply by regulating the amount of enzyme present. One example of this can be seen in HMG-CoA reductase. This is our regulatory enzyme we'll learn about in cholesterol synthesis. HMG-CoA reductase synthesizes cholesterol. And as cholesterol levels within the cell elevate, what happens is this elevated cholesterol will trigger the ubiquitination of HMG-CoA reductase, and ubiquitin will target the enzyme directly for degradation. So the increase in this example of our product will directly target the enzyme for degradation and thereby reduce the enzyme concentration within the cell. So let's finish with a question, and this question would be similar to something we'd expect to see on the discussion board as a pathway problem. And I'll also give you an example of how this question would be answered as a pathway problem. It also may be an example of a case connection that you might see within the course. So if we ask the question, what are the metabolic consequences of changes to the ubiquitin proteasome system? First, we need to know what the ubiquitin proteasome system is. And from our HMG-CoA reductase example, we know that tagging a protein with ubiquitin targets it to the proteasome, where that enzyme or protein will be degraded. And we're asking the general question about changes. So we could envision two general types of changes to this system. One could be an increase in protein ubiquitination, and in this case, this could lead to an increase in protein degradation. In the context of regulating an enzymatic reaction, if we increase protein degradation, this would lead to a decrease in product production. Conversely, if the change leads to a decrease in protein degradation, this could lead to an accumulation of the enzyme or protein product. If an enzyme were to increase within the cell, this could lead to an increase in product production. So these could be the two possible changes we might envision that could be possible related to this question. If we look at a specific example from a publication in 2008, we can explore the rapid degradation of one of the subunits of an enzyme we'll talk about later, the pyruvate dehydrogenase complex. In this paper, they were exploring a mutation which caused the E1 subunit of this enzyme, the pyruvate dehydrogenase complex, to become targeted for ubiquitination. In this case, individuals who had this mutation where the E1 protein was rapidly targeted for ubiquitination and degradation by the proteasome illustrated a deficiency in the pyruvate dehydrogenase complex. This paper also showed that by the addition of a proteasome inhibitor, they were able to alleviate this problem such that the E1 subunit of the protein could then be translocated to the mitochondria and assemble with the rest of the enzyme subunits to generate a functional pyruvate dehydrogenase complex. So this paper is a single example where they were able to show that changes in the proteasome ubiquitination initially caused a deficiency in a protein assembly, and then they illustrated that by inhibiting this process of degradation, they were able to allow for functional assembly of the enzyme. The full article can be found here for anyone that's interested.